Welcome back, learners. In previous part we had discussed about the acid base characters or properties of amino acids, how the amino acid can exist as a Zwitter ion in the solution. Now in this part we will discuss a very important concept of amino acid. How can we separate the amino acids, what is the isoelectric points of different amino acid, and how can we calculate the isoelectric points of amino acids. So first, why does amino acid show Zwitter ion formation? It's very important question. Have you think about it? Why does amino acid show Zwitter ion formation? Because in aqueous solution amino acids are more stable in Zwitter ion forms. Also in solution ion dipole interaction, the charges on amino acids and water, and similarly in solid state the ion-ion interactions between the neighboring amino acids molecules takes place. So sample of amino acids in a solid always present in ionic content. According to Henderson equation, acidic form predominates if the pH of solution is less than the pKa value of compounds. So pH is inversely proportional to the 1 upon concentration of hydrogen ion. When it is decreases, it means that pH is less, and if pH of any solution is less, the pKa value is also less, and if the pKa value is less, the solution will be in acidic form, that molecule will be in acidic form. Similarly, when the pH is more, it means that the pKa value of the solution is high. If the pKa value of solution is high, the solution will be in basic form. So at physiological pH, that is pH equal to 7.3, an amino acid exists as a dipolar or Zwitter ion, have a positive or negative charge on non-adjacent atom. It means that there is no net charge. Few amino acids have side chains with ionizable hydrogen which can exist more than 3 forms. So this is very important point you have to always remember, because they are like histidine. Histidine have a ionizable charge, that side chain. So in that case the amino acid can exist more than 3 forms. Example you are looking here, this is the structure of histidine, and histidine having a ionizable side chain. This is CH3CH-COOH. So this is a ionizable side chain. So at pH 0, that NH3 have a positive charge, but at pH 4 the side chain having negative charge and positive charge both in the side chain. You can see on your screen. At pH 8, again the side chain having both positive charge and negative charge, but at pH 17 only COO minus having O minus charge. So deprotonation takes place when the pH of the solution is reached more than 10. So here side chain is having ionizable hydrogen. So where is the ionizable hydrogen you are looking here? In COH group and NH3 group. So in that case the histidine or any amino acids which are having ionizable side chain can exist in more than 3 forms. Solubility of Zwitter ion is minimum at that pH, and this property is useful to the separate amino acid from the mixture. So the pH at which we get the Zwitter ion, it's always remember in a solution, because we get a cationic form and anionic form also, but at a 1 pH we get a dipolar ion, that is called Zwitter ion. So Zwitter ion at the pH, when the Zwitter ion is get, the pH is minimum, that, and that on that pH, the value of that pH is used to separate the mixture from the amino acid mixtures from the proteins. Now, what is the isoelectric points? We have to understand what is isoelectric points. So, as I told you, isoelectric point is the point, the pH at which there is no net charge. So, we have represented isoelectric point, that is a Pi. So, pH at which Zwitter ion or dipolar ion exists, and at that time the molecule have no net charge, or we can say the electrically neutral molecule, and no movement of ions takes place, no movement of protons takes place. And because there is a no movement of proton takes place at that pH, that is called amphoteric ion. So, Zwitter ion is also called amphoteric ion, dipolar ion. So, the pH at which we get a Zwitter ion or amphoteric ion we call isoelectric point. Example on your screen, you are looking here. Example here, there is a amino acid, and this amino acid structure of glycine. So, always you have to understand, except the glycine, all amino acids having a chiral carbon atom. In here there is a H, in place of side chain we are having H. So, chiral carbon is replaced by any other side chain in any amino acids. So, here at pH 0, because at pH 0 it is a strongly acidic form. So, in strongly acidic form the hydrogen will be more. So, cationic form exits. In higher at pH 0 the pKa value will be 2.3, and the bipolar ion have pKa value is 9.7. Similarly, in anionic form pK3, which I have represented here, pKa3 value, that is the pKa value of first is less than the pKa value of ordinary carboxylic acid. What is the reason? Because due to inductive fact we are having NH3 group. So, that NH3 group create a inductive fact. So, due to that inductive fact of ammonium cation, which help to stabilize the carboxylate ion when it loses the proton. So, the protonated amine group of alpha amino acid as also acidic in nature, but less than carboxylic group, isoelectric point is calculated on the basis of that. So, for glycine we have to calculate it. How we have to calculate the isoelectric point? So, P, for that there is a formula, Pi equal to half pK1 plus pK2. So, in previous slide we have seen the pK value is 2.3 and pK2 value is 9.7. So, that is divided by 2. So, that is the 6. So, 6 is a isoelectric point of alanine. Now, what does it means? It means that when base is added, or when we add the base into the solution, cationic form of alanine at pH 0 takes place and deprotonation started from the carboxylic group. Similarly, during that reaction, when pH reaches at 2.3, the carboxylic acid proton is removed from half of the molecules, and at pH 2.3, from half of the carboxylic acid proton is removed, and at that case, at that time the pKa value will be equal to the pH value. Similarly, when base is added to the solution, pH reacted its isoelectric point, all proton from the carboxylic group removed, but not from ammonium ion proton. So, the ammonium ion proton does not removed from the molecule, and in that case we get a dipolar ion. So, this is the brief explanation why the amino acids exist in Zwitter ion form at their isoelectric point. Similarly, here you are looking here, this is again Zwitter ion, and Zwitter ion adding more base. When we add more base, what happened? The NH3, from the NH3 ammonium ion, it has to started its proton deprotonation has started until the pH of the solution reached 9.7, and half of the ammonium ion will have lost proton. So here adding more base, all H removed from the ammonium ions. So half of the proton from the carboxylate ion removed, and when we add acid and when we add more base, half of the proton from the ammonium ion have lost. So and in between of that we get a Zwitter ion. So at pH 14, molecule carry a net negative charge from their carboxylicate. It means that the anionic form of pH 14, the NH2 is here neutral form, and this is the titration curve which show the equilibrium, and here the graph represent the change in pH as the function of number of molar equivalence of base. As you can see at pH 6, the alanine exists as a Zwitter ion. It is a graph you can see here. As the pH is increases, deprotonation takes place from the carboxylic acid. When concentration of OH is increases, protonation takes place from the ammonium ion, and in middle of that we get a Zwitter ion, and that pH is a 6 pH. So that 6 pH is equal to the isoelectric point. So that 6 pH we get a Zwitter ion of alanine. So both NH2 group protonated and we get a dication, and that dication is strongly acidic. We get that dication is strongly acidic solution. So if amino acids have side chain, now what happens in that case? The carboxylic group and amino group like arginine, histidine and lysine. So all these having a side chains along with the COH group and NH2 group, these molecules having a side chain. So in that case these are the triprotic amino acids, and all can donate or accept a proton because there is a side chain and the side chain also having a proton. So these are the triprotic. As we have seen in the previous example, alanine is a diprotic because only NH2 group and only COH group can deprotonate or protonate, but these are the example of amino acids which are triprotic, and they can all these amino acids can donate or accept a protons. So this is the example of lysine. So here it is lysine amino acid, the protonated from its sigma carbon atom. From where protonation takes place? From the sigma carbon atom. First proton from the carboxylic group lost, and when the pH is raises, in this case the pKa value will be 2.2, and we get a alpha aminium ion, and when we get a alpha aminium ion, the pKa value will be 9. So here up to that we get dipolar ion, and at dipolar ion we get the pKa value will be 10.3. So here cationic form and anionic forms. So how we will calculate the isoelectric point of these? So this is a very important point you have to remember during the calculation of isoelectric points. Isoelectric point also called isoionic point. So isoionic point is the pH at which amino acid does not migrate in electric field. It means that in that time, at that time Zwitter ion is dominant, right? So here acidic medium, low pH value, right? And in neutral form basic medium is high pH. So for diprotic amino acids Pi will be half, pKa plus, pKa2 plus. These are the pKa values. So isoelectric point is halfway between the glycine, as I have discussed here glycine. So in glycine, the previous slide you have seen the glycine having pKa value 2.34 and pKa2 value is 9.7. 6. So it means that the isoelectric point is the halfway between the anionic form and cationic form of amino acids. So it is the 5.96. So for glycine, the isoelectric point of glycine is 5.96. It is nearly 6. So again, if there is a R group which is ionizable or any acidic side chain like aspartic acid having a acidic side chain. Also, if there is a neutral acidic form which can exist under more acidic condition, and the pH at that time is 1.88, between the 1.88 and 3.65. So this is the aspartic acid. It's very important, learners, because aspartic acid having a 2 acidic group, 2 carboxylic group in their molecule, in their structure. So this is the aspartic acid. At low pH we get a acidic medium. So what happen when the concentration of pH of the solution is increases? It means that when the concentration of pH is increases, deprotonation takes place. So both the COH group deprotonated, from both the COH or carboxylic group, proton removed. Similarly, in basic medium, when pH is high, what happen? The charge is neutralized and we get a Zwitter ion. So in that case, what will use? We have to calculate the acidic value of these two form, pKa1 plus pKa3. So we will not consider the pKa2 value. It's very important how to calculate the isoelectric point. So because in aspartic acid, though there is 2 acidic group, so you have to add only pKa1, value of pKa1 and value of pKa3. So that is the 2.77. So the isoelectric point of aspartic acid is 2.77. So isoelectric value is pKa values of 2 groups which is positively charged in acidic solution and uncharged in basic solution. So you have to add only the pKa value of the molecule which is positively charged in acidic solutions. So this is the, again you can see arginine having 2 NH2 groups. So here you have to add the pKa value of the amino group or basic the solution which are having the basic medium, and here in this case you have to calculate not all the pKa values, just you have to calculate here the pKa values which show the positively charge of the nitrogen atom as on the amino groups. So if R is basic side chain or there is extra positive charge, as we had discussed, example of histidine. Here you can see the titration curve over here. So in titration curve we will find out the isoelectric point of this amino acid pKa1 plus pKa3, that is 5.65. So the isoelectric point of histidine is 5.65. So you have to understand, always remember how to calculate the isoelectric point of the amino acid if they have a ionizable or non-ionizable side chains. So this is again you can see here histidine molecules titration graph, and from that titration graph you can easily understand the pKa value of OH equivalent versus pH. Now for calculating isoelectric point, if the side chain of amino acid does not having any ionizable group, so it's very important, always remember, if there is a side chain of amino acid which do not have any ionizable group, in that case the isoelectric point will be average of alpha amino and average proportional to the COH group of values. So we have to add only average of alpha amino and average of that carboxylic group value on these two forms. If side chain has an ionizable group, if there is amino acid which are having a R which is ionizable, all three pKa values must be considered. Understand, it's very important. So in that case you have to add all three values. So if acidic side chain like aspartic and glutaric acid, Pi value will be pKa average of side chain with pKa value of COH in both the amino acids. In case of tyrosine and cysteine, Pi value will be average of middle of pKa value of and alpha COH group, pKa value of alpha COH group. But if side chain is basic in nature like histidine, arginine or lysine, all these amino acids are basic in nature and they are having a side chain which are having NH2 group. In that case the pKa value will be the average of pKa value of alpha value of all the amino acid plus sigma amino acids. Now how to calculate the Pa value of lysine? So it is the lysine structure, you can see here lysine. In lysine you can see here there is a 2NH2 group, right, one on that chiral carbon atom and one NH2 group having a in a side chain. So in that case the Zwitter ion having an extra positive charge, because if the pH is increases the H is decreases, deprotonation takes place from the ammonium ion in the basic medium. So in that case you have to calculate the isoelectric point of both the pH which is a greater 8.95 plus 10.79. So lysine is in a basic in nature because we are getting 10.79. It means that the lysine is basic in nature. Similarly glutaric acid having an extra negative charge because it is having a 2 COH group. If we calculate the Pi value, we will calculate the lower values, pKa lower values. And if basic amines, for basic amines, it's very important point. Always remember for basic amines, it means that basic amines mean that the side chain having amino group, you have to use higher values of pKa. If there is a acidic amino acids, you have to use lower value of pKa for calculating the isoelectric point. Now question is arising, what is the use of these isoelectric points? By using these isoelectric point, we have to find out the structure of amino acids. So here how on the basis of amino acids isoelectric points we can separate the amino acids or we can detect the amino acids from the proteins. So this is a method, electrophoresis. Electrophoresis is a method which we have to use for the separation of amino acid from the proteins. So on the basis of isoelectric points of amino acid, we can separate amino acid. At isoelectric point, the solubility of amino acid is minimal, and at that isoelectric point we get a Zwitter ion. So this is electrophoresis method. Electrophoresis is the motion of dispersed particles relative to a fluid under the influence of especially uniform electric current. So here you are looking here Zwitter ion. So for the movement during the electrophoresis, movement of a protons from cationic and from the anionic takes place. So here how we have to separate amino acids during the electrophoresis. So this is a electrophoresis method. Here we have to take a long strip of cellulose acetate, and in middle of the paper we have to place the mixture of amino acid. So electrophoresis is carried out using paper, starch, polyacrylamide and argosegel and cellulose acetate. Mostly we have to use cellulose acetate as a solid supports. So it is a movement of, during the electrophoresis, movement of charged particles takes place under the influence of electric current. So that cellulose acetate is a thin and uniform micropore structure, chemically pure, and does not contain lignin or hemicellulose or nitrogen. So, by using that cellulose structure, there is a good mobility of amino acids. So, this is the way we have to separate the amino acid from the mixture of from the proteins during use of by using of cellulose strip. So, these are again pi values here. Now, how to detect amino acids? Here amino acid, because after separation of amino acid, how can we find out the amino acid? Because all the amino acids are colorless and crystalline compound, and the certain chemical compound react with amino acid to form a color compounds. Learners, Nin-hydrin test. So, Nin-hydrin test is used to identify the amino acid, which amino acid present in the protein. Now, this is the Nin-hydrin, and all the amino acid except proline gives Nin-hydrin test, because here Nin-hydrin test proline, there is a secondary amino group. So, it gives yellow precipitate, yellow color solution, yellow color in the solution, while all the amino acids which are having NH2 group gives the purple colored complex. So, this is the reaction mechanism. During the reaction mechanism, we get a purple colored complex. This is a Nin-hydrin test, and because the proline is also alpha amino acid, but is having secondary amino group, it gives a yellow brown complex. And this is the reaction. You can see how the reaction takes place with amino acids which are having primary group, amino group, and the amino acid which are having secondary amino groups. So, on the basis of that, we get a blue color if there is an NH2 group, and if there is a secondary amino group, we get a yellow colors. And learner, this is the last point I am discussing here. Nin-hydrin test is used for the identification of the lifting out of fingerprinting. So, forensic sciences also use Nin-hydrin test, because it chemically bond with amino acid in the fingerprint and very good for the lifting prints from paper and cardboards. So, this is the same reaction takes place. The here fingerprint development reactions, here Nin-hydrin react with amino acid which is present in the fingerprint residue, and we get a rumen purple dyes. So, in this lecture we had discussed about the what is the Zwitter ion, how we can separate amino acids, and we have learned there electrophoresis is the best way to separate out the amino acid from the proteins. And after the separation of amino acid, how can we find out the amino acid? So, we have seen there here Nin-hydrin test. Thank you.